From the editors of Biological Psychiatry and the Society of Biological Psychiatry, this is Biological Psychiatry Live, and I'm Dr. Tamara Gore, the social media editor. And I have with me today several authors, and could you please go ahead and introduce yourself, starting with Anke.
Hi, thanks a lot for inviting us. My name is Anke Hüls, and I'm an assistant professor of epidemiology and environmental health at the Wharton School of Public Health at Emory University in Atlanta. And I'm the first author on this paper.
Congratulations and welcome.
Thanks.
Hi, I'm Eliza Wingo. I am an associate professor of psychiatry at Emory, and I am a co-corresponding author on for this article.
Welcome. Dr. Epstein.
Hi, I'm Mike Epstein. I'm a professor of human genetics at the Emory University School of Medicine, and I also have an adjunct appointment in the Department of Biostatistics and Bioinformatics.
Thanks so much for joining us today. And Dr. Wingo.
Hi, my name's Thomas Wingo. I am an associate professor of neurology and human genetics.
Thanks so much, all of you, for being here today with me. If we could start by talking about what is the current, prior to your study, understanding of the role of DNA methylation and cognitive trajectory and cognitive decline?
Yes. So one thing that is novel about our study is that we looked at cognitive trajectory instead of, for example, Alzheimer's disease specific pathologies or clinical diagnosis of Alzheimer's disease, which are the most common outcomes that previous studies looked at in association with DNA methylation. And of note, it is important to say that about 40% of the variance in cognitive trajectory, only 40% is explained by these new pathologies. So that leaves up to 60% unexplained by new pathologies in cognitive decline. So there is really a need to better understand cognitive trajectories and also basically what is going on in the brain when people start having a severe cognitive decline before they even have any kinds of neuropathologies. That's what our study did.
So you're really focusing on the percentage, it sounds like the majority of cases which don't have obvious neuropathological links, and asking the question of what is the role for DNA methylation in those cases. Is that correct?
Yes, so our main outcome was cognitive decline. And we also conducted several analyses in which we specifically looked at individuals who had a steep cognitive decline, but they only had low or moderate signs of neuropathologies. And our study showed that even in these patients or individuals who had no or only little signs of neuropathologies, we still found associations with DNA methylation in brain that could not be explained by these changes in neuropathologies.
That brings me nicely to my next question, which is what is the new understanding of the mechanisms underlying cognitive decline that your study uncovers?
Yeah, so we found an association between differential DNA methylation of sites in the Claudin 5 locus and cognitive trajectory. And this association between DNA methylation and CLDN5 and cognitive decline was, as I said, even significant in participants with no or little signs of Alzheimer's disease neuropathologies. And this really could suggest an early role of CLDN5 and the related blood-brain barrier dysfunction in cognitive decline.
That is very novel. And what also is novel is the method that you use to identify this association relative to traditional methods for epigenetic analysis. Can one of you describe the innovative features of your approach and why you think it assists in gene identification?
Sure. So traditionally in epigenetic analysis, the traditional sort of strategy for analysis is to look at the relationship between one CPG site and one phenotype, sort of a univariate comparison. But what we know is that in many situations, many genes are pleiotropic. That is, they can influence multiple distinct correlated phenotypes, together in a pleiotropic fashion. And we have multiple measures here of sort of various correlated cognitive domains that we can sort of jointly analyze. By jointly analyzing them, we can have better power to detect signals. The additional thing that we can also consider here is the idea that within a gene, we would expect CPG sites that are close to one another within a gene to have correlated methylation profiles as well. And leveraging that correlation can also improve power relative to the univariate sort of techniques. So we took methods that my lab had previously developed for sort of genome-wide association analysis or next generation sequencing analysis, where we look at multiple genetic variants in a gene and compare it to multiple phenotypes that we found leads to improved power in that setting. And we applied it here in the epigenetic setting by swapping out the study of multiple genetic variants in a gene with multiple sort of CPG methylation profiles in a gene to gain power that way.
Were any of you surprised by the finding and the identification of Claudin 5?
I was personally. It certainly, what's interesting about this and sort of neat about the collaboration is that not only do you have the multivariate sort of approach, harnessing the correlation within the CPGs within the gene, but also the outcomes. And so the outcome here is cognitive trajectory. And what we were, as Anka already alluded to, we're looking at things that are sort of independent of what are traditional AD measures. So these are brain pathology measures that have been used in a variety, or at least 3 or 4 other studies. And they implicated a variety of genes. And at least the prior studies really focused exclusively on neuropathologic outcomes. Mm-hmm. And for obvious reasons. And so taking this approach, we find in cognitive trajectory, especially people that are more rapidly declining, a gene that is integral into the blood-brain barrier formation being associated with more rapid decline. Mm-hmm. And that certainly is very intriguing from sort of a neurobiology standpoint and sort of the recent work understanding the neurovascular unit, and also speaking to the potential for vascular compromise mm-hmm. as an important initial step in some degenerative diseases, not only Alzheimer's, but the vascular cognitive impairment and others that have it really identified through imaging analysis, for instance, the important role that the vascular integrity seems to be associated with, or the lack of vascular integrity, I should say, associated with more cognitive impairment in a sort of additive fashion to, say, that of the traditional pathology of Alzheimer's disease. So it ties together 2 different sort of fields in a very unexpected way, in my view, at least.
Yeah, to add to what Thomas said, I think what's also pretty unique about this study is that we looked at cognitive trajectory, which gives you a lot of information. So let me explain what cognitive trajectory is. It is basically imagine a line on an x-axis and a y-axis. So essentially cognitive trajectory is something that we have to obtain by following the participants over time, mm-hmm. assessing them every year. So we have data points longitudinally. Mm-hmm. So if you have more than 2 or 3 data points, and in this cohort, I think we have usually at least 5 or 6 data points, you can establish a trajectory where it will tell you whether the cognitive performance over time is stable, mm-hmm. or is it improving, which is unlikely, or it's declining. And it also tells you the rate at which the cognitive performance is improving or declining. So this is possible by longitudinal study, which is rather unique and rich in the information. So cognitive trajectory give us not only the rate. So when we talk about decline, we know that someone was performing within normal range, and then they are diagnosed with mild cognitive impairment or dementia. So we know that there's decline in cognitive performance, but we don't know like the rate of it. So that's why this study is pretty unique. And
Yeah.
the information was based on objective testing.
Yeah, there's certainly a lot of novel features to your study. My last question for you is, what clinical implications, if any, are there for the methylation of claudin 5 and the blood-brain barrier more broadly in cognitive decline and dementia, if you were willing to hazard a guess?
Well, I think that the ultimate.
Please speculate wildly.
Sure.
This is your chance to speculate well.
Will do. I think that the goal or the finding of vascular related protein or gene certainly was unexpected, but certainly fits well into the existing framework, as we kind of already alluded to. And I think that the narrow focus of the field of Alzheimer's work on neuropathologic accumulation is well justified, but we probably need to think a little bit more broadly too, that there are probably additional factors that are adding to cognitive burden. And what Eliza, I think, was alluding to as well is that, you know, the rates of impair or rates of decline and the fact that people do decline are really clinically what's most relevant. Whether somebody has a specific diagnosis in practice, I find people rarely care that much about what, you know, specific kind of dementia or whether they have MCI. What they really want to know is, am I declining? Is this worse than what we would expect? And I think that the identification of an early change or this change and that may be acting in combination would certainly open the realm, or would certainly suggest that we should study biomarkers that are sensitive to vascular compromise and think of a model that is more additive rather than an either or, which is often the way things are sort of explained now. Like, well, there's an accumulation of PIB positive or the radio tracer that binds to beta amyloid, for instance, or some of the tau radio tracers, that these things are not acting only in isolation. And that's what David Bennett, who's conducted the large Ross map study that we were sort of analyzing, their work also speaks to that. So taking it to the next level would be devising biomarkers or something along those lines that would assess the integrity of the blood-brain barrier as a independent set of sort of understanding of cognition. And I think the other element of this is that, like we often suggest in medicine, you should get a screening colonoscopy at such and such an age. That what this study also highlights is that the decline over time is what's maybe more relevant, or at least needs to be considered in addition to an absolute threshold that says, okay, if you are below, you're not remembering, you know, you have 10 items and you remembered 3. Now that's a problem, where if you remembered 5, everything's fine. You know, when I sort of say it like that, obviously it makes it sound absurd, but the idea is that we really do have a very rigid sort of comparison against a group mean. And that probably is not as applicable to some people, as, especially as we think about how to screen for a cognitive decline. We should consider annual assessments that include some element of these tests that we could then apply as people get older, that are when they're asymptomatic. So to me, that I think it helps identify that there's a need both clinically to consider asymptomatic screening, similar to how we do for what we approach a lot of other, you know, screening for blood pressure, diabetes, etc., colonoscopy, and in addition to the consideration for blood-brain barrier modalities for new biomarkers.
I think this study is it's a tour de force and certainly lays the groundwork for that. So congratulations. And thanks again for taking your time today to talk to me. I really enjoyed it. I enjoyed our discussion. Thank you.
Well, thank
Thanks
you.
a lot.
Appreciate it.